the audience. We'll move on to our next talk, who is Elli Brimble, who's a genetic counselor, who most recently was a clinical assistant professor of neurology and co-director of neurogenomics program at Stanford. She has a master's of science degree in molecular genetics from the University of Toronto, as well as her genetic counseling degree from Boston University. She's on the scientific board of FOXG1 Research Foundation, as well as Syngap Research Fund. Importantly, in the context of this setting, she's the head of clinical operations at Ciitizen, managing transformation of unstructured clinical documentation into structured data sets. So Ellie, we're looking forward to your talk.
All right, can everyone hear me okay?
Yep, we can hear you and we can see your slides.
So first off, thank you so much for inviting me to speak today. I'm looking forward to share with you some of the work that we have started in partnership with SRF. So unsurprisingly, I am a full-time employee of Ciitizen. And the agenda for my talk today is to give you an introduction to Ciitizen if we haven't already met before. I'm going to overview our pipeline, as well as the scope of work that we have planned with SRF. And then at the end, I wanted to present a sample data extraction. And the purpose of this is really to give you some sense of the data that we're collecting, and to start you thinking about how that data might be useful for some of the work that you're leading individually. So to start off with, Ciitizen is a patient-mediated software as a service platform that allows users to collect and store their medical records. And then our process is to turn those medical record documents into computable, digital, and relevant data sets that can be shared at the user's direction. Everything on our platform starts with the patient. So we have a very cute little girl here. Either the individual themselves or their caregiver onboards onto our platform and can place a request through the app. We then leverage the HIPAA right of access to go and collect their medical records on their behalf. And this will include not only the medical record documentation, so you can see here I have a very small example of a progress note and a genetic test report, but we also collect the full DICOM imaging studies as well, all in efforts to facilitate other research. Once we receive these medical record documents, our core process is really working to develop a machine learning and natural language processing assisted extraction method. But of course, everything goes through at least one layer of human review. And ultimately, the output is this structured, encoded data set that can be easily shared. When we think about generating real-world data, it's really critical that we approach data collection in a systematic and consistent manner, but also that we're taking consideration into what kind of data we collect to ensure that it's relevant. So at Ciitizen, we approach this in two different ways. One is with our disease model. And so this really serves as the conceptual foundation for our data extraction. It's defining what kinds of data we take and also the relationship between those data entities. So we're not necessarily creating some linear roadmap, but really a complex network of data that demonstrates the relationship between the various elements. Underneath the disease model, we have our manually curated ontology. And the purpose of this is to supply the disease model with standardized terminologies. And this is critical for harmonizing and normalizing the multiple data sources that we collect for each individual, and ultimately will support data portability. This slide is a little bit overwhelming and is certainly not one that I anticipate anyone to memorize, but it's really just to give a sense of the types of information that we systematically collect across individuals. This is our disease model. Each of these boxes outside of patients represents a type of data that we collect. So for example, in the context of SYNGAP1, the primary diagnosis would be a SYNGAP1 related disorder. And you can see that there's an arrow connecting the primary diagnosis with the biomarker because as we know, Syngap is achieved using genetic testing. And so there's a connection between the genetic test results and the resulting diagnosis. Importantly, we also collect clinical diagnoses, which is sort of an umbrella term that we use to reference clinical phenotypes and features. Some of these diagnoses may have been arrived at using a diagnostic procedure. Some of them may be receiving treatment in the form of a medication or procedure. And then there are a handful of other data elements that we determined we wanted more detailed information about, so they actually have their own section. And this primarily includes things like seizure history, exam findings, and development for the most part. So overall, we have this standardized and systematic approach to data capture that we've tried to make comprehensive, but really focusing on those endpoints that we think will be most useful for rare neurologic disease in the context of developing appropriate therapies and identifying and characterizing endpoints. This is just a brief snapshot of our ontology. So as part of our data extraction, we kind of go through, identify relevant elements within the source text, but everything is filtered through this ontology to apply that layer of normalization. And we use 3 internationally recognized terminologies in the ontology, primarily SNOMED for clinical phenotypes. So here you can see this was kind of an earlier iteration of the coordination section of our physical exam finding entity. So these are all a number of standard terms, each with a SNOMED code assigned to it, meaning that we're talking about the same things in the same way every time. We use RxNorm primarily for medications. Importantly, there's an ingredient model computed here, so we're able to recognize when brand and generic medications actually contain the same ingredients. And then LOINC we primarily use for measurements. And so in this case here, you're seeing some examples of lab values. Putting it all together, as I mentioned, we start really with this unstructured source text, which as most everyone on this call knows, represents our most rich data source. And we go through and identify relevant information, but then filter those through the ontology. So here's an example of a neurology exam. And what I'm showing you here is how we can really pull out specific pieces of information from that exam, filter them through the ontology, and apply this layer of standardization to the data that we extract. In terms of our scope of work plans with SRF, we will be onboarding at least 100 individuals with SYNGAP1 related disorder. We will perform the medical record collection as well as our systematic data extraction. And then as I've just described, all of that data is fit to our disease model as well as the ontology. And then ultimately, we're also working towards being able to partner this data with patient-generated health data, primarily in the form of surveys, diaries, and validated measures. But of course, we're interested and excited to see what we can do with wearables in the future as well. So again, moving into the pilot data collection, this is not meant to share any new earth-shattering news about SYNGAP, but really just meant to give you an overview of the data that we collect and some of the applications of that data. So we randomly identified 10 individuals who have become citizens as part of this effort to generate real-world data for SYNGAP1 related disorder. And I'm going to show a few examples of some of the data that we extracted from these individuals' records. So just to start with, the mean age of the 10 individuals was 8.8 years and ranged from 3 to 18. We had a relatively even distribution of males and females with 6 females and 4 males. In terms of the age of diagnosis, the mean age for the cohort was 5.7 years, but ranged from 1.8 all the way up to 15.1 years. And unsurprisingly, all diagnoses were achieved using next generation sequencing, primarily clinical testing. In terms of medical record collection, just to orient you to this slide, the graph that you see here is showing the number of documents collected across the cohort by year. As you just saw, most of our patients were underneath 10 years of age. So it's unsurprising that most of the medical records were collected later on. But importantly, we know that institutions only need to save records for 10 years. So it could also explain why in general we tend to see fewer documents before 2010. In terms of the requests generated by the patients when they onboarded to the platform, they're able to generate two different types of requests. One are medical records, and the second is radiology. So we received an average of 5.8 requests for these 10 individuals, ranging from 2 to 12. And this roughly equates to institutions. So this would be a mean of about 6 institutions per individual. And then radiology requests were an average of 4.1, ranging from 1 to 9. And then in terms of what we actually received, we received on average 100 documents per individual, ranging from 20 to 203. And this equated to around 1,000 pages of documents per individual, ranging from 132 all the way up to 3,881. Part of our process is to break down these sort of larger aggregate documents into individual document types. And so you can see here that by far and away, the most common document type that we received for Syngap, and this is really true across the board outside of Syngap as well, is progress notes with a total of 669 across the 10 individuals. And then you can see here the frequency of the other types of reports that we collect as well. So overall, we had a fairly comprehensive medical record collection for these 10 individuals with an average of 6 providers per person. Most of these represented progress notes, which are really our best data source for the type of information that we extract. And then one thing that I just wanted to point out is that once we collect the records, the individuals or their guardians have direct access to these records and are able to share them quite easily. So it represents an immediate benefit to the participants as well. In terms of the variance observed in the 10 individuals in this cohort, we had almost a roughly even split between frame shift, missense, nonsense, and intronic variants, with no overlap in the individual variants identified. And in this case, all missense variants were confirmed to be de novo. We know Syngap 1 is primarily thought of as a loss of function disorder, so we were able to confirm that those with missense had either a likely pathogenic or pathogenic classification. Most of these were achieved through exome sequencing, but there were a few panels as well in here. One of the primary data points that we extract are what we call clinical diagnoses, but as I mentioned earlier, this really just represents clinical features and phenotypes. And so we extract these systematically across the documents that we receive. And for this here, I took the more specific terms and mapped them back to a system. And so that's what I'm showing in this table here. The middle column represents the total number of unique diagnoses that map to that system. And the percent column here is showing the proportion of individuals with at least one diagnosis that maps to that system. So we can see overwhelmingly the most common features reported fall within this sort of umbrella category of neuropsych, with all individuals having at least one diagnosis falling into this category. This was followed by neurology with fewer unique diagnoses, but again, a high proportion of individuals with a neurologic diagnosis. And then the third most frequent were musculoskeletal findings, with about 30% of the cohort having at least one. So looking at the more specific terms, these were the features that were identified in more than 50% of the cohort. And you can see here that it maps fairly closely with what we just heard from Dr. Khani. So most commonly reported was hypotonia in 90%. On the far right-hand corner here, I'm using the earliest time this phenotype was described in our medical records as a proxy for age at diagnosis, but I do expect this to become more accurate with more individuals. Right now, this is really just more for your information that we collect this in this type of data rather than thinking of this truly as the age of onset. The second most common were autism spectrum disorder and global developmental delay. And then epilepsy was documented in 7 individuals. And then here I wanted to demonstrate again that these are normalized and standardized terms, each of which are mapped to a specific code. When we looked at features that were present in more than 25%, but less than 50%, again, we're seeing a large number of neuropsych and neurologic features, so intellectual disability, anxiety, autistic features, constipation, regression, language disorder, sleep, and speech. One thing I wanted to point out here is that we do anticipate that some features are likely underreported. So two that stand out to me here might be constipation or sleep disorder. I would anticipate that sleep in particular would have been reported much higher. One of the benefits of our approach to data capture is that we build in a little bit of redundancy. So even though we're seeing a lower frequency of sleep disorder or a sleep problem reported than what I would anticipate, because we also collect things like medication, we can cross-reference to see if there are any medications that are most likely being used to treat sleep to better understand the frequency of this in the cohort. And that is a good segue into talking about medication. So as I've alluded to, we do systematically extract medication use across documentation as well. So in this table here, we counted the number of unique medications used in these 10 individuals. 18 were used to treat epilepsy, 5 for behavior, 2 for sleep, 1 for anxiety, and 1 for GI. On average, there were about 8 unique medications documented for each individual, and on average, about 5 epilepsy medications were documented in the 7 individuals who had epilepsy. Um, Note that these are not currently being taken at the same time. This is just historically the unique medications per individual. And then I actually just added this in response to one of the questions. In these 10 individuals for this pilot, 5 reported use of either the ketogenic diet or a modified Atkins diet in efforts to treat their epilepsy. One of the benefits of our extraction is that we pair every data point to a specific date. And so this allows us to look at things by age as well, which is really just what I'm demonstrating here. These are the number of medications that each individual or that was reported across the cohort per age. And so you can see that there is some variability, but the average is ranging between 0 and 2 for the most part across individuals. But it did range pretty significantly from 0 medications per year all the way up to 11 medications per year, depending. So again, depending on your specific research questions, there's a lot that we can do with the data that we collect. One of our entities, as I showed earlier on, is Diagnostic Procedures. So here I pulled out the findings that were reported for EEGs in particular. So within the cohort of 10 individuals, 9 had documentation of at least one EEG. And on average, each individual had 3.9 EEGs, and this ranged from 0 to 9. On this table here, I'm showing the most frequent findings that were reported on the EEG. Generalized epileptiform discharges were most frequent, with seizures being captured on EEG as a close second. And the most commonly reported seizure types were primarily generalized seizures, so absence, atonic, and myoclonic seizures. And then you could see here there were a number of other findings that were reported in more than one individual as well, including things like background findings, as well as normal EEG findings as well. We also collect developmental outcomes. So the 5 here that I presented are some of the heavy hitters that we talk about more frequently, but we do have fairly comprehensive ability to capture more specific milestones when they're mentioned. So things like running, jumping, academic skills as well, like using numbers, recognizing colors, ability to read, etc. So in this case here, we're showing the frequency of individuals that were reported as being able to perform that skill. as well as the age at which that skill was documented as being attained. So we can see about 70% of individuals were able to sit, achieved at a mean age of 12 months. 80% of individuals were reported to walk independently, achieved at a mean age of 22 months. 20% were reported as able to use a pincer grasp, but again, this may be reflecting less common reporting of pincer grasp use, but the mean age here was 39 months. 60% were reported as being able to use at least one word, achieved at a mean age of 27 months. And then 20% were reported as being able to use short phrases for a mean age of 28 months. In the cohort, we had no documentation of wheelchair use, and 2 individuals were using a communication device. And then the last thing I wanted to share is an example of some of the data we collect from physical exam findings, in particular neurology exam findings. So we extract pertinent positive and negative findings across cranial nerve, muscle bulk and strength, muscle tone, sensation, reflexes, coordination, and gait. So within this cohort of 10 individuals, we documented 79 unique neurology exams. This was an average of 6.1 exams per year across the cohort, or an average of 8 per individual. And just based on some of the findings that we know are more common in Syngap, I wanted to show some examples of findings that were frequently reported for muscle tone, coordination, and gait. And so for all 3, you can see that actually the most frequent finding for these exam or for these individual components of the neurology exam were actually normal findings. But we do see, particularly for muscle tone, there were frequent reports of low muscle tone. We did see decreased coordination, clumsiness in coordination reported at times for the coordination component of the exam. And then we also identified some abnormal gait findings as well. Across these 3 components, 80% had at least one abnormal muscle tone finding on exam, 60% had at least one abnormal coordination finding, and 70% had at least one abnormal gait finding. And then lastly, I did just want to highlight some limitations of this approach. We know that our data extraction is going to be limited to what's actually present in the medical record documentation. So there may be omissions and there may be inaccuracies. We're actually able to mitigate the inaccuracies fairly well by focusing our capture on the specialties that we think are most pertinent to the entity that we're extracting. But omissions are a little bit harder for us to correct for. One way that we do try to achieve this is by collecting across institutions. So we're not limited to a single hospital or a single practice. As you saw, we were actually collecting records from a large number of institutions per patient. And so this is one way that we try to target omissions. But also importantly, for many of these rare conditions, we don't really have standardized clinical guidelines yet. And there will be variable access to expert medical care. And so we are going to see some inconsistencies in clinical management. And so the volume and type of data available across patients will be equally variable as well. And then there also are some data elements that are probably going to be better suited to patient-generated health data. So whether that's something like a validated measure of sleep or behavior or a seizure diary, we're really looking forward to be able to pair the patient-generated data with our objective data from the medical records to develop the most rich and comprehensive data set that we can. And so with that, I'd be happy to take any questions.
Great, thank you, Ellie. I think that's a really exciting platform and quite remarkable, actually, that you can pull all that information from the records. So there are some questions. Let's see here, where should we start? Are you able to generate a view of changes in the data over time? For example, to model changes in epileptiform activity over the patient's life?
Yes, so I didn't do that here for the sake of time, but because all of our findings are linked to a date, we can actually report everything out based on the age of the patient. So we can model things like EEG findings, exam findings. We can watch how those things evolve over time. Either, hopefully we're hoping to be able to get a large enough cohort that we might be able to do that in a genotype specific way, but certainly across the cohort in particular, we can.
That would be great. Another just general question about the platform, not Syngap one specific. When you pull out medication names, is that information automatically paired with the condition it's being used for? So for example, as a psychiatrist, some medication used for epilepsy are also used for mood stabilizers, and unless it's documented, that can be confusing.
So we actually purposely do not assign things automatically for the exact reason that you just said. We know that there are various reasons why these medications may be used, and particularly in pediatrics, some of these may be off-label. So we have a field where we can pull in the indication for the drug, but we only use it when it's specifically commented on by the prescribing clinician. As a result of that, not all of our medications that we record will have an associated indication, because that is one of the omissions that we see in the documents. But we kind of recognize that as a limitation, and map back to the treatment indication as best we can based on that type of medication.
Got it. And then another question about medications is someone asked how often patients got valproate as an anti-epileptic compared to others. So I think frequency of treatment with that medication compared to others is the question.
So I was just going to say, I actually have it pulled up and could chat it after, but I don't think it will necessarily be that meaningful in a group of 10 patients.
Yeah.
But that is absolutely something that we can pull out and will be much more meaningful when we have a larger group to present.
Great. And then Janie was asking about the balance between adding in family surveys to basically to supplement the information, I think, that you get from medical records. Yeah, and then trends over time, which I think you sort of answered.
Yeah, so one of the things that we're really excited about at Ciitizen, which is a little bit outside the scope of this talk, so I apologize, but we kind of like the idea of building this toolkit that advocacy groups can use to launch their own research. And one of those initiatives is to be able to arrive at a standard set of either free or very cheap validated measures that can be used to assess phenotypes and symptoms that we know are important in neurodevelopmental disorders. So things like sleep and behavior and seizure frequency. So those are all things that we're planning to be able to offer soon. And then the frequency at which those surveys are administered would really be up to the advocacy group or the research partner to determine how often they want us to be sending those things out, keeping in mind the burden on the families as well.
Great. Will you be able to correlate mutation types to clinical presentation and history?
Yes.
Great. I like that answer. And then finally, what machine learning, natural language processing techniques do you use, and do you have any preliminary results about how accurate these techniques are?
Yeah, I'm going to do a terrible job at answering this question, so please feel free to follow up and I can connect you with our ML team. I don't know any of the technical terms, but I think active learning sounds familiar to me. Basically, one of the things we do is we generate a ground truth data set. So there's sort of always this reference that is used to train, and then the machine actually trains itself within the data as we're using it. So part of our work right now with SRF is to develop that ground truth data set, as well as our measures of precision recall, which are then used to generate an F1 statistic. And that is our readout of accuracy. So we started in cancer at Ciitizen, and we have that data for cancer, and we're working on building it right now for neurology.
Great. And Mike, it looks like you might have a question or comment.
I have a couple, unfortunately. Ellie, could you give an update on how many records have been collected, how researchers get their hands on this data, You didn't talk about how many Nomad codes you've made, which I think is so cool because it suggests that you guys really are trailblazers. And could you just talk about what other groups are getting involved?
Yeah, of
And by the
course.
way, thank you. It's been a real pleasure working with you on this.
Oh, anytime. Likewise, likewise. So I'm really sorry. I don't know off the top of my head the number of records that have been collected, but we have 50 patients signed up. I think we're basically done record collection for them. So 50 times an average of 60, or sorry, of 6 would give you your number for about the number of records we've collected for these individuals. And then as Mike will probably share with you, we're about to onboard another 50 as well. One of the really important things for us as Ciitizen is that we never want to be a barrier for research. So all of the data that we're generating is available to academic research or nonprofit use with a license that is essentially free. So if at any point you think this data would be useful to further your own research, please reach out and we can work to start being able to supply that data to you. In terms of the other groups that we're partnering with, we have a smaller cohort of individuals with mitochondrial DNA mutations on the platform. We have another smaller cohort of individuals with SLC13A5 deficiency, which is one of the autosomal recessive epileptic encephalopathies. We are about to onboard FOXG1 syndrome onto the platform, and we're particularly excited about that because we just received one of the CZI Rares 1 grants to help build out the ML component for neuro with not only FOXG1, but also Syngap research and SLC13A5 deficiency as well. And then I'm not sure if I'm allowed to talk about anyone who's not on the platform yet, but we have a lot of people coming on in 2021 too, which we're excited about.
Nomad Codes.
Oh yes, Nomad Codes. So I am turning into a Nomad nerd, so I really apologize and hope you'll accommodate this. But Nomad is so cool. One of the coolest things about it that I didn't talk about is that it stores hierarchical relationships. So even though we are accounting for a lot of redundancy in the way to talk about things because we want to be comprehensive, ultimately we can walk all of those codes back up to a common denominator, for lack of a better word. But one of the things that we were realizing with SNOMED is that it really wasn't tailored towards pediatric rare disease. And so part of our efforts at Ciitizen, one of our directors is a member of some sort of international SNOMED working group. So we have been steadily expanding our ontology to accommodate for some of the more common findings in pediatric in general, but in particular pediatric neurodevelopmental disorders. And so I think we're up to about 250 new SNOMED terms that we've generated. And these are actually going to be submitted to the SNOMED working group. So we're basically the community at large can use these. And when they are implemented into SNOMED, they will be using the codes that we created. So we're not sort of siloing our information. We're making sure that it's accessible to anyone that uses SNOMED.
Great. I think we've hit all the questions and we're right on time. So thank you, Ellie. That's an exciting platform and project.